Hello everyone. Welcome to SS EDUCARE Teaching Learning channel. Thank you for all my subscribers. And for those who didn't subscribe my channel, please do subscribe and watch my videos. In my today's topic, I am going to teach part 2 of chapter 2, cell cycle, cell division and structure of chromosomes. Let's go to our channel. Story of the day. Once a girl went to her father and said her life is full of miseries and every day she is facing so many struggles in life. She is not interested in life, it seems. In her young age itself, all the troubles and all the struggles are coming only to her. She started complaining that life seems to be very boring and she is not interested in this life as she is not having happiness. She is having only the miseries in life, unhappiness throughout her. Her father did not reply for her. Instead, he took her to the kitchen and asked her to keep 2 pots on the stove. He then asked her to pour some water in the pots and asked her to boil it. The water started boiling. In the boiling water, he asked her to drop potato in one pot and egg in the another pot and asked her to keep an eye on it for 15 minutes. After 15 minutes, he asked her to watch the potato and the egg. Since it is boiling, she couldn't take it out. After some time, he asked her to take it out and tell about it. She took the boiled potato in one hand and boiled egg in the other hand. As her father asked her to say about it, she told that potato has become soft and the egg has become hard. Then the father started telling about it. Father told that, see, the same boiling water, same temperature, same time of heating, the potato became soft and the egg became hard. Initially, the potato was hard. It became soft. But the egg was very fragile. Now it became very hard. See, for 15 minutes of boiling, continuous boiling, the same potato and egg has faced so many hardships. And finally, it has come to a shape that it cannot be easily broken by anything. Isn't it? The complete taste of potato will be felt by us once when it is becoming soft. And the complete taste of egg will be felt by us when it is becoming hard actually. That is what is the story for today. Moral of the story. In life, things happen to us, things happen around us. But we should not be worrying for all those little things. We should keep learning, keep adapting for the struggles and convert our struggles into positive deeds. Today's topic, I am going to teach part 2, meiosis of cell cycle, cell division and structure of chromosomes. The next cell division is meiosis. Meiosis is also called as reductional division. This meiosis, calling as reductional division, was coined by J.B. Farmers in the year 1905. Why is meiosis called as reductional division? Because instead of the chromosomes getting double, the chromosomes are reduced into half. So, meiosis is called as reductional division. The cell undergoes 2 successive divisions, immediate divisions, that is one after the other division, and forms 4 daughter cells in meiosis. Among these 4 daughter cells, each daughter cell contains half the number of chromosomes from its parent cell. See here, parent cell has diploid. Diploid means it is 2n. The number of chromosomes is 2n. Whereas in the daughter chromosomes, we are getting only haploid. Haploid means it is n condition. Parent is diploid 2n condition and the daughter ones are haploid n condition. That is why meiosis is called as reductional division. This meiosis occurs in germ cells. Mitosis occurs in somatic cells. But meiosis occurs in germ cells. The germ cells are also called as reproductive cells. For male, they are the sperms, and for the female, it is in the ovum. This meiosis includes two divisions, meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. The number of chromosomes is reduced into half in case of daughter cell, so it's called as reductional division. We already discussed. Then it is also called as heterotypic division. As we said, meiosis 1 is called as heterotypic division. What is this heterotypic? Heterotypic is nothing but the formation of daughter cells with half the number of chromosomes. Reducing the number of chromosomes, we call the type of division as heterotypic division. Continuing, the cells which undergo meiosis are called as meiocytes. Meiosis 2 is also called as homotypic division. Meiosis 1 we said as heterotypic because hetero means different. What is different over there? The chromosomal number is different. It is getting reduced. Whereas here homo means the same. The chromosomal numbers are remained same. There is no change in the number of chromosomes in meiotic 2 division. That's the introduction for meiosis. Now, first meiotic division, meiosis 1 will be discussed. Each chromosome replicates into two sister chromatids. Each chromosome, one arm above and one arm below, is replicated, duplicated. As already I have shown the diagram previously, duplicating is exactly the forming of Xerox copy of itself. One above and one below. Thus it forms two sister chromatids. The homologous chromosomes come together and pair during prophase 1. The homologous chromosomes, homo means the same, similar type of chromosomes, they come together and pair during prophase 1. Each pair of homologous chromosomes containing 4 sister chromatids are called as tetrads. I will draw the diagram later, but each pair containing 4 chromatids are called as tetrads. Since it has 4 arms, we are calling them as tetrads. Meiosis 1 has 4 phases. Prophase 1, metaphase 1, anaphase 1 and telophase 1. The next is homologous chromosomes. Previously, I told homologous chromosomes, they have same loci. So, this homologous chromosomes are the set of chromosomes which have one from father and one from mother. They pair up with each other inside a cell. They are called as homologous chromosomes. So, one chromosome from father and one chromosome from mother pairing up to form homologous chromosomes. Next, they have the same set of genes at same loci. So, what is this loci? Loci is the position where genes are found. You cannot find the genes throughout the chromosomes. You can find the genes in a particular places which are called as loci. Look at this diagram. This is one paternal and maternal chromosomes. Paternal means paternal means from father. Maternal means from its mother. So both the chromosomes have the same loci. same loci. Then they also have similar length of arms and centromere position. Look at this diagram. The length of the arms are the same and the position of centromere is also the same. That's why they called as homologous. Logous means position. Homo means same. Next topic is crossing over. The non-sister chromatids of tetrads. Previously we have seen if a chromosome has 4 arms, we call them as tetrads. The non-sister chromatids of tetrads seems to cross each other at a specific point called as chiasmata. The points or the place where 2 arms of chromosomes or chromatids cross over each other is called as chiasmata. The next is non-sister chromatids. What do you mean by non-sister chromatids? Look at this. As I already said, 1 chromosome from father and 1 chromosome from mother forms a tetrad like this. See, paternal chromosome, maternal chromosome. When 2 such chromosomes cross each other, see, I'll tell you what are sister chromatids. The arms of them are called as sister chromatids. That is, a daughter from same parent, father and mother, same parent, they are called as sister. As how they are called as sister, they are also called as sister chromatids. But the arms of this chromosome and the second chromosome, when they cross each other, these 2 are called as non-sister chromatids. The arms of the same pair, the same homologous pairs are called as sister chromatids, and the arms of this one and the arm of this one, these 2 are called as non-sister chromatids. When such non-sister chromatids undergo crossing over, when they cross against each other, the place of their crossing over is called as chiasmata. Look at this place, it is called as chiasmata. Next, at this chiasmata point, the 2 chromatids break down and rejoin again, forming new recombinant chromatids. Thus forms new chromatids with maternal and paternal chromatids. See, they are the fresh maternal and paternal 2 sets of chromosomes. They undergo crossing over like this. And they form, see, look at this, you can find the difference. They have been broken down and rejoined here, forming recombinant chromatids. And the one that doesn't undergo crossing over called as non-recombinant chromatids. The next topic is significance of crossing over. Why should crossing over occur and what is the importance of crossing over? It forms the basis of genetic variations in organism. What do you mean by genetic variation? Let me give an example. One side I have hibiscus plant, which has an ability to give many number of flowers. On the other side I have rose plant, which has an ability to give different types of flowers, different colors of flowers. When these 2 are being crossed over, the resultant plant will have both the characteristic features, that is, giving different colors of flowers and many number of flowers. So, the genes are varying, differing from its parent cells. So, that is what is genetic variation. Nowadays, genetic variation is much more helpful in the market. Next, it also helps in mapping of chromosomes. Mapping means locating the genes in the chromosomes. Take for example, all the chromosomes seems to be similar. We wanted to know the specific type of gene. Suppose in that chromosome there is a crossing over region or chiasmatic region. If the genes are there, we can easily locate so and so gene is present in that particular chiasmatic region. So therefore, X gene is present in X chiasmatic region. So this crossing over region is helpful to locate the genes clearly. Next, second meiotic division. or meiosis II. In meiosis II, two daughter cells undergo division to form 4 haploid daughter cells. In the first division, I said the number of chromosomes are reduced into half, whereas here it's not been reduced, it is remaining the same. Two daughter cells are forming 4 haploid daughter cells. Then, this meiotic division has 4 phases: leptotin, zygotin, pachytin, diplotin, and ending up with diakinensis. Coming to leptotin stage. Leptotin stage, the chromosomes, they align, they move and then prepare for recombination. Look at this. The chromosomes, they move, they start moving apart and prepare for recombination. Next stage is zygotin. Zygotin is the stage where crossing over takes place. So, the chromosomes are coming close so that one of its arms are ready to cross over. Next, pachytin stage. Chromosomes separate. After crossing over, chromosomes tries to separate from each other. See, chromosomes are getting separated in pachytin stage. Next, diplotin stage. Diplotin. So, chiasmata break down and the pairs move apart. In the diplotin stage, see, the crossing over region or the chiasmatic region, they separate and the chromosomes move apart. In the final stage, that is diakinensis, the crossed over chromosomes pairing up, they pairs up in the last stage, diakinensis. See, look at this, the chiasmatic chromosomes or the crossing over chromosomes, they paired up and form the homologous chromosomes. Next, the difference between mitosis and meiosis. Mitosis occurs in somatic cells. Somatic cells, as I already said, they are bodily cells, the cells that are present in the organs of the body. Meiosis occurs in the germ cells, which are also called as reproductive cells. For male, it is sperm, and for female, it is ovum. Mitosis produces cells for growth and repair. It undergoes division for the purpose of growth and for repairing of cells. Meiosis undergoes division for producing male gamete and female gamete since it is during reproduction. Mitosis, the 2 daughter cells are formed, whereas in meiosis, 4 daughter cells are formed. Each daughter cell is diploid 2n, whereas in meiosis, each daughter cell is haploid. Since it is reductional division, 2n is reduced to n, haploid condition. Next, in mitosis, the daughter cells are identical. They look similar. They are the same. Whereas in meiosis, they are genetically different because it undergoes crossing over. We have seen non-sister chromatids undergo crossing over and form chiasmatic chromosomes. In mitosis, no such crossing over occurs. Thus, in my next video, I am going to teach structure of chromosomes. That will be the last part of this chapter, cell cycle, cell division and structure of chromosomes. Thank you for supporting me.